Hello and welcome to the Biological Anthropology lecture series. I'm Dr. Richard Jonathan O. Tadurin. Here we explore what it means to be human, biologically, evolutionarily, and behaviorally. Let's begin our journey into the science of humanity. Welcome to Module 2, the biological basis of evolution. Here we explore cells, DNA, inheritance, and the evolutionary processes that shape all life, including humans. In this section, we explore population genetics, Hardy-Weinberg equilibrium, and the forces of evolution that drive genetic change. Let's begin by zooming out from individual genes and looking at how entire populations carry and transmit genetic information. In biology, a population isn't just a group of individuals living in the same place. It's a community of interbreeding organisms that share a common gene pool. That gene pool, the sum of all alleles in that population, is the level at which evolution truly operates. Let's look at how we describe variation in that gene pool. Instead of focusing on genotypes of single individuals, population genetics looks at allele frequencies. You might wonder why we shift to this mathematical view. The reason is that allele frequencies give us a measurable way to see whether evolution is happening. If the frequencies stay the same over generations, the population is stable in a genetic sense. If they change, evolution is underway. This brings us to the Hardy-Weinberg principle. Think of it as a baseline model, a way of saying, if nothing is acting on this population, here's what we expect the genetic makeup to look like. The principle states that allele and genotype frequencies will remain constant from generation to generation in the absence of evolutionary forces. Hardy-Weinberg isn't telling us how real populations behave. It's telling us what no evolution looks like so we can detect when something is causing change. Let's walk through the basic idea. Imagine a gene with two alleles, P and Q. If no evolution is occurring, the genotype frequencies in the next generation should fall into predictable proportions. P squared for homozygous dominant, 2pq for heterozygous, and q squared for homozygous recessive. What's powerful about this model is that we can compare real population data to these expected frequencies. If they match, the population may be in equilibrium. If they don't, something is pushing it away from equilibrium. You might wonder what kinds of things push populations away from that idealized state. Migration can bring new alleles in. Mutation introduces new variation. Natural selection changes the success of particular genotypes. Genetic drift causes random shifts, especially in small populations. Non-random mating alters how alleles pair up. Each of these forces changes allele frequencies, which means evolution is taking place. Let's look at an example to make this concrete. Suppose a population has a known proportion of recessive phenotypes for a trait. By taking the square root of that frequency, we can estimate Q, the recessive allele frequency. From there, we can calculate P and then the expected genotype frequencies. When we compare these predicted values to observed data, we can see whether the population behaves as the Hardy-Weinberg model predicts. If not, we've identified evidence of evolutionary forces at work. Keep in mind that population genetics is the link between molecular variation and evolutionary patterns. It gives us the tools to understand how small genetic changes, multiplied across generations and populations, shape the diversity we study in biological anthropology. Let's take a deeper look at the forces that actually drive evolution. First, let's revisit the idea that evolution is fundamentally about changes in allele frequencies across generations. These changes don't happen randomly or all at once. Instead, they arise from different processes, some predictable, some chance-driven, that act on populations over time. Understanding these forces helps us make sense of why populations look the way they do and how they continue to change. Let's start with mutation, because it's the ultimate source of all new genetic variation. Think about DNA replication as a process that is incredibly accurate but not infallible. Occasionally, A base is copied incorrectly, or a small segment gets inserted or deleted. Most of these changes are neutral, some are harmful, and once in a while, a mutation creates a new allele that may later become advantageous. You might wonder why such rare events matter. The answer is that evolution is a long game. Even tiny changes can accumulate and become meaningful when they occur over many generations. Now, let's look at natural selection, the force we usually think of when we hear evolution. Selection acts on variation that already exists in the population. Individuals with traits that improve survival or reproduction tend to leave more offspring, shifting allele frequencies toward those advantageous variants. But selection doesn't push all traits in the same direction. It can be directional, favoring one extreme, stabilizing, preserving the average, or disruptive, favoring two extremes at once. A classic example is the sickle cell allele in regions where malaria is common. The allele is harmful in the homozygous state, but heterozygous individuals gain protection against malaria. This balancing act shows how complex the relationship between genes and environments can be. Let's turn to gene flow. Gene flow happens when individuals migrate between populations and reproduce, mixing alleles from different gene pools. Think about human history. Populations have always moved, interacted, and exchanged genes. When gene flow occurs, it tends to reduce genetic differences between populations. You might wonder why this matters for understanding human variation. The answer is that gene flow creates connectedness across human groups. Many patterns once attributed to long-term isolation vanish once we consider how often humans have moved and mixed throughout history. Now, let's explore genetic drift. Drift is evolutionary change driven purely by chance. Instead of favoring traits that are beneficial, drift reflects random fluctuations in allele frequencies. These effects are especially strong in small populations. Imagine flipping a coin only a few times. The results can easily skew toward heads or tails. But flip it thousands of times, and the ratio levels out. Populations work the same way. A rare allele can become more common or disappear entirely by random events alone. One dramatic form of drift is the founder effect, which happens when a small group breaks off to form a new population. The genetic diversity of that new group depends heavily on who happened to be in the founding group. Another is the bottleneck effect, where a large population is suddenly reduced, often by natural disasters or disease. The surviving population carries only a small fraction of the original genetic diversity, and that limited gene pool shapes future generations. Let's bring these forces together. Mutation introduces new variation. Natural selection filters that variation based on environmental pressures. Gene flow moves variation between populations. Genetic drift reshapes variation through random processes. All four forces interact, often simultaneously, creating the dynamic patterns we observe in human populations. Always remember that evolution isn't a single event or a linear process. It's a constant interplay of forces acting on genetic variation. These forces help explain everything from global patterns of human diversity to localized adaptations. Understanding them gives us a clearer view of how populations change, survive, and evolve, and why humans today are both deeply connected and remarkably varied. Let's revisit what we've uncovered in this section. We shifted our focus from individual genes to whole populations, and in doing so, we learned how allele frequencies, rather than individual traits, tell the real story of evolution. Population genetics gave us a framework to measure that story, while Hardy-Weinberg equilibrium offered a baseline for recognizing when evolutionary forces are at work. Let's look at how the different forces came together. Mutation supplied new variation, natural selection filtered that variation, gene flow mixed populations, and genetic drift reminded us that chance can play just as strong a role as adaptation. Now, this part is important. None of these forces acts in isolation. Real populations are shaped by combinations of these processes, often at the same time. That's why patterns of human diversity are complex, because our evolutionary history is a tapestry woven from movement, adaptation, history, and randomness. You might wonder how these abstract concepts translate into the bigger questions of biological anthropology. The answer is that every pattern we study, whether it's global variation in height, disease resistance, or genetic markers, reflects the long-term action of these forces. Population genetics helps us interpret those patterns, allowing us to see whether they emerged through adaptation, migration, bottlenecks, or simple drift. As we close this section, keep thinking of populations as dynamic, interconnected systems. By understanding how evolutionary forces shape allele frequencies, we gain a clearer picture of how humans have evolved and how populations continue to change in response to shifting environments and ongoing human movement. Now let's revisit where this module has taken us. We began at the microscopic level, looking at cells, DNA, and chromosomes, the structures that store and transmit biological information. From there, we followed that information outward, seeing how genes are expressed and how small molecular events give rise to traits. And finally, we zoomed out even further to populations, where those traits interact with environments, movement, chance, and history. Let's look at the big takeaway. Evolution is not a single event or a simple progression. It's a continuous, multi-layered process that starts with DNA replication, unfolds through gene expression, and ultimately plays out in populations over generations. You might wonder why this matters when we move into later modules on primates, fossils, or human variation. The reason is straightforward. Every pattern we observe, from the shape of a hominin femur to the distribution of blood types across continents, depends on the mechanisms we explored here. Now, this part is important. Understanding the biological basis of evolution gives us the tools to interpret both continuity and diversity. It helps us see why humans share so much genetically, yet also show remarkable variation. It explains how adaptation emerges, how populations respond to environmental pressures, and how genetic histories are written into our bodies. As we close this module, keep thinking about evolution as a story written at multiple scales: molecular, individual, and population. In the next modules, we'll build on this foundation and begin tracing that story across deep time through our primate relatives and the fossil record of our own species. Thank you for joining this lecture. I'm Dr. Richard Jonathan O. Tadurin. I hope this session deepened your understanding of biological anthropology. Keep exploring what it means to be human. See you soon.